So how does your cochlea encode frequency of sound, right? Which will ultimately perceive, as sounds of different pitches. Well, the cochlea is, this coiled structure, right? It has a base end where the stapes is sort of poking in, right? And then it coils all the way down, right, to the tip or the apex, the end point here. And, running down the middle of that coil is a membrane called the basilar membrane. And the basilar membrane is, where the hair cells actually sit. And, the inner hair cells that are our primary sensory neurons for, hearing, with free-floating hairs in the fluid inside the cochlea, the endolymph, right? And also the 3 rows of outer hair cells, which are involved in sort of motor or efferent, adjustment of the, the physical characteristics of the cochlea in different auditory environments. So it changes the acoustics, right, of our cochlea, how they're responding. So the basilar membrane is key to the tonotopic organization, or the frequency mapping, of the cochlea, because at the base end, where the stapes is again sort of poking in and out, over here, I guess, poking in and out over here, right? It's narrow. The basilar membrane is narrow and stiff. So it's very narrow and stiff. So if you have high frequency sounds, right, it's gonna wobble, the narrow, stiff base end of the basilar membrane in the cochlea, and you're going to move the inner hair cells like this. You're going to splay open, the tips, pull on the tip links, right, and you're going to open channels that allow for a depolarizing inward current, which in the case of the inner ear, right, is weirdly carried by potassium, not by sodium like it is almost everywhere else. Basically, as the basilar membrane, continues from that base end all the way out towards the apical end or apex, it gets wider and sort of floppier and more responsive and more, mobile in response to sounds of lower frequencies. So the lower frequency sounds are going to actually displace a different part of the basilar membrane, something further along towards the apical end, which will move those hair cells. And these are the ones that are going to be, sort of responding to lower frequency sounds. So there is a tonotopic organization or frequency mapping. The location of the inner hair cells along the basilar membrane matters. Those near the narrower or stiffer base end actually vibrate in response to high frequency sounds, while those near the wider, floppier sort of apical end are jostled in response to lower frequency sounds. This is a place code, right? It's dependent on the place that gets displaced, right? A tonotopic organization. The topography of the basilar membrane is based on the tone or the frequency of the sound.